Welcome to the deep dive. We're absolutely thrilled to have you with us today.
So glad to be here.
For this exploration, our mission is to really look at the blueprint of the mammalian little brain.
Right, the cerebellum.
Exactly. And uncover what exactly makes the human brain so, incredibly unique.
It's a profound question and one that scientists have been trying to answer for literally centuries.
Yeah.
To do that today, we're going to be diving into a genuinely groundbreaking 2024 study. It's by Sepp and colleagues, published in the journal Nature.
A huge publication.
Oh, absolutely. The study is titled Cellular Development and Evolution of the Mammalian Cerebellum.
And the sheer scale of the research we're looking at here is, it's just staggering. I mean, the researchers sequenced the RNA of around 400,000 individual cells.
400,000?
Just to trace the development of the cerebellum.
Yeah.
But before we get into the weeds, let's clarify what that actually means.
Good idea.
Sequencing the RNA of 400,000 cells sounds impressive, but what are the researchers actually looking at when they do that?
Right. So think of it like, imagine trying to understand how a massive skyscraper is built.
Okay.
Instead of just looking at the finished building from the outside, single-cell RNA sequencing allows you to basically intercept the daily work orders.
Oh, wow.
Yeah, the work orders and text messages of every single one of the 400,000 construction workers on the site.
So you see everything.
Everything. You can see exactly what instructions they are receiving, what tools they're turning on, and what specialized job they're doing at any given microsecond.
That is wild.
By doing this, the researchers track the brain's construction from early neurogenesis, which is when the brain is just this tiny cluster of forming cells, straight through to adulthood.
And they didn't just look at humans, which makes this so fascinating. They compared human brains to those of mice and even to the marsupial possum.
Which is such a specific choice, yeah.
Right. By mapping these three lineages, they were essentially looking back across 160 million years of mammalian evolution.
Mind-blowing.
But it begs the question of why the cerebellum is the star of the show here. Because when most of us picture human intelligence, we immediately visualize the neocortex.
We do.
That highly wrinkled, walnut-looking outer layer of the brain.
The neocortex definitely gets all the Hollywood glory, for sure.
Right.
But the cerebellum, which sits tucked quietly at the very back of the skull, is the unsung hero of our biology. It actually contains more than half of all the neurons in your entire brain.
Wait, really? Let that sink in for a second.
Yeah.
This tiny structure holds the vast majority of your brain's processing units. More than half the brain's neurons crammed into the back of the skull. That completely shifts how you have to think about brain power.
It changes everything. And historically, the medical field completely pigeonholed this massive neural network.
How so?
Well, we thought of the cerebellum as just the motor control center, the hardware that helps you balance on a bicycle or coordinate your hand to catch a baseball.
It's just movement.
Exactly. But this study, alongside a whole wave of modern neuroscience, underscores that it is vital for so much more.
Like what else?
It is deeply involved in advanced cognitive processing, affective and emotional regulation, and even linguistic processing.
Language?
Yes. You literally cannot have human-level cognition without the immense computing power of the cerebellum.
Okay, let's unpack this. To understand how this complex, densely packed little brain comes together, I want you to imagine a highly organized, massive construction site.
I love this analogy.
Right, because the development of the cerebellum doesn't just happen in a random biological soup.
No, not at all.
It relies on three main germinal zones. And you can think of these zones as specialized manufacturing hubs on our construction site, with each hub dedicated to producing specific types of cellular workers.
That is the perfect way to visualize it. If you want to really memorize how this works, let's walk through those three manufacturing hubs one by one.
Let's do it.
The first hub is the ventricular zone, or VZ. I like to think of this as the brake factory.
The brake factory, okay.
Mm-hmm. Yes. This is the birthplace of GABAergic neurons. Now, in neuroscience, when you hear GABAergic, which stands for gamma-aminobutyric acid, just think of the brain's red lights.
Red lights, got it.
These are inhibitory neurons that act as the brakes in the brain's electrical circuitry, stopping signals from just spiraling out of control.
So the ventricular zone is pumping out the brakes.
Right.
What kind of specific cells are rolling off that assembly line?
Several key players. It produces deep nuclei neurons, interneurons, and the famous Purkinje cells.
Purkinje cells, I've heard of those.
They are incredible to look at under a microscope. If most neurons are like, say, tiny little shrubs,
Yeah.
a Purkinje cell is like a massive, beautifully branching ancient oak tree.
Wow.
They act as the main output of the cerebellar cortex. But the VZ also produces some highly specialized cells that actually pack their bags and migrate completely out of the cerebellum into the brainstem.
They leave the site entirely.
They do. These become the parabrachial and neurogenesis neurons.
Let's ground those terms really quickly because they sound heavy. What do those migrating neurons actually do for us on a daily basis?
They keep you alive and awake.
That's pretty important.
Very. Parabrachial neurons help regulate critical survival functions like your breathing rate and your heart rate.
Okay.
And the neurogenesis neurons are heavily involved in your fight-or-flight response and alertness. They are the chemical alarms that wake you up and tell you to pay attention to your surroundings.
So our first hub, the VZ, handles the brakes, the massive oak tree Purkinje cells, and the survival alarms. What is the second manufacturing hub?
The second hub is called the early rhombic lip, and this is associated with the nuclear transitory zone, or NTZ.
So if the first hub makes the brakes.
The second hub makes the gas pedals.
Okay.
It gives rise to glutamatergic neurons. Glutamatergic simply means excitatory. These are the green lights that push electrical signals forward.
So glutamate equals gas pedal.
Exactly. These gas pedal neurons assemble at the NTZ and include glutamatergic deep nuclei neurons, which help route information, as well as extracerebellar isthmic nuclei neurons.
Which brings us to the third and final hub on our construction site.
That would be the late rhombic lip, associated with the external granule cell layer. Think of this as the mass production plant.
Mass production, meaning they make a lot of something.
Tons. This area is entirely dedicated to churning out granule cells, which are actually the most numerous neurons in your entire brain.
Billions of them.
Exactly. It also produces a unique type of cell called unipolar brush cells, or UBCs.
I was actually looking at the notes on those unipolar brush cells, and there's this alphabet soup of genetic markers mentioned.
Right, the genetic markers.
The researchers found a whole new hidden subtype of these cells, and they tracked it using a genetic marker called HCRTR2, right alongside a known marker called EOMEs.
Yes.
From a purely scientific perspective, why is finding a hidden cell subtype like that such a big deal?
It is a huge deal because it proves how much detail is still hiding in our own biology.
Okay.
Imagine you've been studying a busy factory for decades, and you think you know every single type of worker there.
Right.
Then, by looking closely at their individual tool belts, in this case, by sequencing their single-cell RNA and finding that HCRTR2 marker, you suddenly realize there's a highly specialized class of clumber you never knew existed.
Oh, that's a great way to look at it.
It just goes to show that even in structures we thought we fully understood, zooming into the genetic level reveals hidden layers of diversity. And amazingly, this newly discovered plumbing crew is conserved across mammals, meaning nature decided it was crucial millions of years ago.
Let's just pause here for a second to make sure we've got the foundation set, because that was a lot of incredible architecture to visualize.
It is a lot.
So here is a quick recap. Number one, your cerebellum is built from 3 distinct manufacturing zones. You've got the ventricular zone making the brakes, the early rhombic lip making the gas pedals, and the late rhombic lip acting as the mass production plant.
Spot on.
Number 2. Incredibly, whether you are an opossum, a mouse, or a human, that basic factory layout has remained practically identical across 160 million years of evolution.
Unchanged.
Which brings up an interesting question for you, the listener, to chew on. If the factory layout is exactly the same across these species, which of these 3 cellular manufacturing zones do you think plays the biggest role in making your brain distinctly human?
That is the perfect question to launch us into the developmental timeline, because this is where the differences between a mouse, an opossum, and a human finally start to reveal themselves.
Okay.
But to compare these species fairly, the researchers couldn't just use chronological age. They had to align their developmental stages biologically.
Meaning you can't just compare a 1-week-old human to a 1-week-old mouse.
Right, because a 1-week-old mouse is practically paying taxes and running its own life.
Whereas a newborn human baby is essentially a helpless potato that can't even hold its own head up.
It's a hilarious but deeply accurate biological truth. Humans are born incredibly underdeveloped compared to other mammals.
We really are.
So, based on the transcriptomic data, meaning the actual RNA activity inside the cells, The researchers found that a newborn human's cerebellum most closely resembles that of a 1-week-old mouse.
A newborn human equals a 1-week mouse.
Yes, and a 3-week-old opossum. When they aligned the timelines this way, they found that the biological sequence of how the brain develops and the precise order in which these cellular hubs turn on and off are fundamentally the exact same across all 3 species.
Here's where it gets really interesting. While the sequence of cell generation is basically identical, the quantity of the cells produced and the timing of when they are produced differ wildly in humans.
Wildly.
We take the exact same blueprint, but drastically alter the building schedule.
We absolutely do. And the most striking difference the researchers found centers on those massive, beautifully branching oak trees we mentioned earlier.
The Purkinje cells.
Yes, the Purkinje cells. At around 8 to 9 weeks post-conception, so very early in human fetal development, human embryos have an approximately 2-fold higher proportion of Purkinje cells compared to mice and opossums at their corresponding developmental stages.
Double the amount.
We are churning out double the amount of these specific complex cells.
That feels like a massive evolutionary upgrade. But in the grand scheme of a brain with billions of cells, why does doubling the Purkinje cells early on matter so much?
If we connect this to the bigger picture, Purkinje cells don't just sit there acting as passive wiring.
What do they do?
They actively regulate the amplification of granule cell progenitors.
The mass production workers.
Right. Basically, they act as the foremen on the construction site who send the chemical signals telling the factory to hire more granule cells.
Okay.
Because humans generate this massive 2-fold boom in Purkinje cell foremen early on, it sets the stage for an explosive exponential generation of the tiny granule cells later in development.
So it's a cascading effect.
Precisely. And this massive increase in total cerebellar volume is mathematically required to match the evolutionary expansion of the human neocortex.
So the neocortex couldn't work without it.
We talk about the wrinkly cortex giving us higher thought, but your cortex simply couldn't process complex human language or deep emotional regulation if your cerebellum hadn't scaled up its own underlying computing power to support it.
And what's fascinating is that it's not just a general across-the-board increase. The study notes that this Purkinje cell boom in humans is specifically biased toward what they call early-born Purkinje subtypes.
Early-born, yes.
These specific early-born cells project their neurological wiring to a region called the lateral deep nuclei.
Specifically an area known as the dentate nucleus.
Right. And from an evolutionary standpoint, the dentate nucleus is exactly the piece of hardware that expanded so dramatically in primates and humans to support advanced cognitive functions, rather than just simple motor tasks.
It is a brilliant example of nature's efficiency. Evolution didn't reinvent the wheel. It just kept the factory running longer, specifically pumping out the early-born foremen to build a much larger, more capable engine connected directly to our higher cognition centers.
Let's do another quick check-in here to synthesize what we just covered.
Sounds good.
Number 1, developmental timelines and construction sequences align beautifully across mammals, but humans have uniquely scaled up the production phase.
We really scaled it up.
Number 2, humans produce twice as many early-born Purkinje cells at a critical fetal stage.
8 to 9 weeks.
And number 3, this Purkinje boom triggers a massive generation of granule cells down the line, driving the sheer computing power needed to support our advanced intelligence.
It's all connected.
So my question to you, the listener, is, how much of your complex cognitive ability, your capacity for language, for art, for reasoning, do you think is owed simply to a prolonged mathematical growth phase of this one specific cell type?
It really reframes how we think about human exceptionalism, doesn't it?
It really does.
It's not always about having completely unique alien biology. Often it's just about proportion. But that being said, there are profound differences at the molecular level.
Which brings us to the software running inside these cells.
Exactly.
Yes, the software versus hardware analogy. I think this is one of those powerful ways to understand this entire study.
It really simplifies it.
The researchers found that the hardware of the cerebellum, the actual physical cell types like the Purkinje foremen and the granule cell workers, is highly conserved across mammals.
Same hardware.
But the software, meaning how, when, and to what degree specific genes are expressed inside those cells, has been rewritten significantly over millions of years.
What's fascinating here is that we are looking at orthologous genes.
Let's define that.
Orthologous simply means these are genes that are shared across humans, mice, and opossums. We all have the exact same basic genetic code sitting in the nucleus.
Okay.
But during the course of evolution, these shared genes have evolved entirely new expression trajectories.
Explain what an expression trajectory is in this context.
Think of a trajectory as a software schedule.
A software schedule.
Yeah. In a mouse, a specific gene might be programmed to turn on for 2 days, reach a medium intensity, and then turn off forever.
Got it.
But in a human, that exact same shared gene might have an updated software schedule that tells it to turn on for a month, reach maximum intensity, and pulse on and off repeatedly.
So same gene, completely different instructions.
Right. The researchers found that in our granule cells specifically, there is a relative excess of these trajectory changes uniquely in the human lineage.
The study highlights a few very specific examples of this.
Yeah.
And rather than throwing a dozen acronyms at you, let's focus on just one to really see how this works.
Good call.
A gene called SNCAIP, synuclein alpha interacting protein. The study noted that this specific gene evolved the strongest changes in its expression trajectory during human granule cell differentiation.
Its software schedule in humans is radically different than its schedule in mice.
Why does a shift in one gene schedule matter so much?
Because a glitch in that specific software schedule can be devastating. This isn't just a fun trivia fact for evolutionary biologists. This has profound clinical and disease implications for all of us.
How so?
When researchers look at genes with shared dynamic expression in both Purkinje and granule cells,
Like the MML2 gene they mentioned in Purkinje cells.
exactly, they find these genes are heavily linked to severe neurodevelopmental diseases.
Like what?
Things like cerebellar malformations and movement disorders like spinocerebellar ataxia.
And returning to that one specific gene, SNCAIP, the researchers point out that it is frequently duplicated in a condition called group 4 medulloblastoma.
Yes.
For those who aren't familiar, that is a highly aggressive and devastating childhood brain tumor. So a gene that evolution fundamentally reprogrammed in humans is the same gene that, when it malfunctions, drives pediatric cancer.
It's sobering.
They also looked at other genes that have human-specific software shifts, genes functioning in granule and Purkinje cells, and found strong links to autism spectrum disorder and intellectual disability.
Right. Genes like MYT1L and KNSL1 in granule cells and SMRCR2 in Purkinje cells.
Which is huge for understanding those conditions.
It is exactly why understanding these evolutionary software updates is so critical. And this raises an important question regarding how we develop medical treatments.
Because of animal testing.
Right. For decades, the biomedical field has relied heavily on animal models, particularly mice, to understand human diseases and test new drugs. It is the gold standard of pharmaceutical research.
We test a compound on a mouse, and if it cures the mouse, we move it to human trials.
But here is the massive caveat this research exposes.
Let's hear it.
If these critical disease-linked genes are operating on a fundamentally different software schedule in humans compared to mice, it means our mouse models might not reflect all the disease manifestations we see in human patients.
Ooh.
A drug that perfectly fixes a cellular pathway in a mouse might do absolutely nothing for a human child with a neurodevelopmental disorder.
Simply because the human cells are utilizing those genes in an entirely different, highly evolved pattern.
Exactly.
That is a sobering thought. It means we might be looking for human cures using a biological map that only partially applies to us.
We have to be aware of those limitations.
Now, before we wrap up this segment, I want to share a fun evolutionary fact from the study about species-specific novelties.
I love this part.
While we mostly share the same hardware and tweak the software, the researchers did find a few completely unique additions.
Brand new code.
Right. They found that human mature granule cells express a gene called ZP2. What's wild is that this specific gene expression was acquired roughly 7 million years ago.
Which is just after the evolutionary split between human ancestors and chimpanzees.
Exactly. And human interneurons express a gene called CPLX4.
Which emerged exclusively in the great ape lineage.
So while we are largely running on upgraded opossum hardware, we do have a few completely custom pieces of software installed in our brains.
It's the perfect finishing touch on the evolutionary picture. A foundation shared with an opossum, scaled up dramatically like a skyscraper, running heavily modified scheduling software.
With a few exclusive high-end human apps installed right at the very end.
Beautifully said.
Let's do our final quick synthesis for this section.
All right.
Number 1, Humans and mice share the exact same cellular hardware, but the software, the patterns and schedules of gene activation, are entirely different.
Completely distinct.
Number 2, these uniquely human gene patterns are strongly linked to human-specific presentations of diseases like medulloblastoma, autism, and intellectual disability.
Which challenges how we study them.
And number 3, certain gene expressions, like ZP2, are entirely exclusive to recent human ancestors and don't appear in mice at all.
Right.
Which brings me to our final question for you before we wrap up. If our brains use the exact same genes in completely different ways than the animals we study in labs, how should we rethink our entire approach to testing neurological treatments in the future?
It is a profound dilemma, and one that the entire fields of neuro-oncology and psychiatry are going to have to grapple with over the next decade.
For sure.
Techniques like single-cell RNA sequencing are quite literally forcing us to reevaluate our baseline assumptions about translational medicine.
So what does this all mean? If we step back and look at the grand narrative we've explored today, from Sepp and colleagues' deep dive into the cerebellum,
It's quite a story.
the real magic of the human brain isn't about nature throwing away the old blueprints and inventing entirely new types of cells from scratch. It is so much more elegant than that.
Much more efficient.
Evolution took a perfectly good 160 million-year-old factory layout. It tweaked to the timing of the construction phases, scaled up the numbers in a massive Purkinje cell boom to amplify our computing power, and fundamentally repurposed the genetic software schedules to build an exponentially more complex neural network.
We are variations on an ancient theme.
But those variations make all the difference in the world.
And that evolutionary theme leaves us with a truly compelling idea to ponder as we close out today's exploration.
What's that?
I want you to think about this. If our cerebellar cellular makeup is fundamentally the exact same as a tiny marsupial opossum's, just tweaked in its timing and its gene expression trajectories, what hidden cognitive capacities might currently be lying completely dormant in our own human genetic code?
Just waiting.
Just waiting for another slight evolutionary shift in gene expression to unlock them.
What an incredible thought to end on. Thank you for taking this deep dive into the blueprint of the mammalian little brain with us. Until next time, keep exploring, keep questioning, and keep diving deep.